Hi students, this is Mr. Eckert and we're going to go through the last target in this first section of targets on cells. This is target C7, which says that you should be able to explain how advancements in technology have influenced our understanding of cells. So of course when you think of technology and cells, probably the most important piece of technology that we can think of would be microscopes. And when you look at the early microscopes, this is an example of one of those microscopes that looks like it was a photograph taken of the microscope in a museum, perhaps. But they really weren't very good compared to, of course, our modern microscopes. They had relatively poor resolution. Resolution is, if you think of your phone and when you take a photograph and look at that photograph, resolution refers to kind of the sharpness of the image. So, with a phone, that refers to like the number of pixels that are in the image, how many megapixels it fits in a certain area. So an older phone might have maybe a 3 megapixel resolution, whereas some of the newer phones might have 8 or 10 megapixels. And that refers to how clear that image. If you zoom in on an image that was taken by a camera that has poorer resolution, then the image you'll see start to get blurrier. Probably a great example or a best example of that is if you look at an image of a photograph that was in the newspaper from, far away, maybe a foot or two distance, the image looks pretty good. But as you get up close, you start to see the individual dots or pixels that make up that image, so it has pretty poor resolution. So some of these early microscopes had pretty poor resolution. Um, it says here that in 1660, Marcello Maffigi, an Italian scientist, he was actually able to see individual blood cells moving in the capillaries of the tail of a live fish. And this is actually pretty cool. If you want to open up the PowerPoint and click on this active link, it'll take you to a YouTube video where you can see this. It's pretty remarkable seeing the individual red blood cells moving through these little capillaries. So, and then it says within the next decade, Robert Hooke and Anton von Leeuwenhoek, two names that should be familiar to you from the previous targets, they saw cork cells and protists using simple light microscopes. So a simple light microscope, the light of course passes through one or more lenses to produce an enlarged image of the specimen. These earliest microscopes only had one lens, whereas the microscope like that you're going to use in class, of course, it's a compound microscope. The light passes through an objective lens and then through the ocular or eyepiece lens. So even with these microscopes, they're pretty limited into how clear they can make an image. So most of the organelles that scientists know of now, they weren't even able to see those with these earlier microscopes. The nucleus was certainly visible, but beyond that, there wasn't a whole lot more that was visible. And that continued until electron microscopes were invented, and that was in the mid 1900s. So instead of using light, which limits magnification to about 2000 power, and the microscopes that we have in class, the the most powerful ones, some of them that have 4 lenses in their revolving nose piece, the biggest lens is a 100 power lens, so that goes up to 1000 total magnification. So the most powerful light microscopes, you're going to see up to about 2000 times magnification. Electron beams, or electron microscopes rather, they use beams of electrons rather than light. And so because of the shorter wavelength of those, electron microscopes can multiply several 100,000 times bigger than the actual image. So one of those that you should know for test purposes is what's called a transmission electron microscope, or TEM for short. And they're actually going to shoot a beam of electrons right through a thin slice of the specimen, and they have to stain the specimen with metal ions in order to do that. So that kills the specimen. I'm slicing it, a thin section of it. Between that and then staining it with metal ions, that kills the specimen. So here you can see one of those Golgi apparatus. You can see the stacks of membranes that make up the Golgi, and then these vesicles that it's producing as it modifies substances within the cells. Here, what do you think this is? This is taken with a scanning electron microscope. And a scanning electron microscope, rather than passing electrons through the object, it bounces the electrons off the specimen and then onto a fluorescent screen. So that, as you can see, is the surface of a cat's tongue. You can see why, if you've ever been licked by a cat, it kind of feels like sandpaper. But this, again, because the specimen has to be coated with metal in order for the scanning electron microscope to pick up the image, then the specimens in this are no longer living as well. Finally, scanning tunneling microscopes, they use a probe, a very narrow probe. It looks like a needle, and it's measuring voltages that are leaking out from the surface of the specimen, leaking electrons, really. That's what it's picking up. So this is the only one out of the three that can actually look at live specimens. So the first two, the specimen is not alive. This was the only one that allows them to look at live specimens. And you can see here, these are actual atoms that were arranged on this plate to spell out, of course, IBM. So these are atoms of xenon, and that's on a nickel plate. So there's 35 xenon atoms that are making up IBM. And that's it for that target. Again, if you have any questions over this target, please make sure you ask.